Here we have a DNA replication fork. The orange dot represents the origin of replication. There is only one in prokaryotes, and it is determined by a series of DNA sequences. It allows for the enzyme helicase to start unzipping the parent DNA strands, like a zipper. The green dots represent single-stranded binding proteins that bind to the newly separated parent strands to keep them apart so the parent strands can be used as templates to synthesize the new strands, sort of like a door stopper, making it easier for movement to work on certain jobs. Gyrase is a topoisomerase used to keep the strands from supercoiling by working ahead of the replication fork to relieve tension on the rest of the DNA. In this example, the twists on the outside of the replication fork get tighter, which is where gyrase is applied to relieve stress. The yellow dots represent RNA primase that play an important role by placing RNA primers, the yellow lines, on both strands as flags to inform the DNA polymerase III where to begin building the new DNA strand, since it must have a 3' end to attach to in order to add nucleotides to the primer and to be able to synthesize the new DNA strands. Here, the purple dot represents DNA polymerase that moves along the parent strand in the 3' to 5' direction, but synthesizes the new DNA in the 5' to 3' direction. This strand is known as the leading strand because it replicates continuously. The strands are anti-parallel, therefore DNA polymerase on the bottom blue parent strand basically builds backwards, creating discontinuous replication and why we call it the lagging strand. The orange strands represent new DNA, and as you can see, there are gaps or fragments between them. These fragments are created from catching up and are called Okazaki fragments. The yellow dots represent RNA primers, which are what the DNA polymerase I, the pink dots, bind and remove to replace RNA with DNA nucleotides. The sparkly yellow dots represent the enzyme ligase, which glue the Okazaki fragments together and fill nicks by using phosphodiester bonds that connect the sugar phosphate backbone to create a continuous strand.